Hello everyone. The paper that I'm going to present next is titled An Easily Accessible Microfluidic Chip for High Throughput Microalgae Screening for Biofuel Production. In this paper, a microfluidic device was developed to optimize the analysis of microalgae culture parameters like density, organisms growth, and lipid content. The most important interest of microalgae, especially in this variety, which is Botricoccus brownie, is the high content of dry matter and the high percentage of lipids, because it's related to the capacity to produce biofuel. In materials and methods, they use the culture of this microalgae to produce it in optimal conditions by themselves. Related to the chip design and fabrication, they use and develop a microchip microfluidic hang and drop device. This device have 2 layers of polystyrene, made with polystyrene. The most important part of the chip, in the middle of this chip was made of PDMS, and the walls that made with this design, they will prepare 3 different diameters to measure the cell growth. The first figure that we can watch here, you can see the three layers. This third layer was made also with polystyrene, but they use opaque black polystyrene sheet because they want to watch to find the influence of the light on this microalgae. So they put a dark region and the region with light. So they use another layer. In this case, we can watch the device and the distribution of the wells. To determine the microalgae growth, they measure the intensity of the chlorophyll autofluorescence every 2 days for 14 days in total. They use this method because the chlorophyll is strongly associated with biomass production. And in the case of the lipid, they analyze through the use of Nile red fluorescent ink, and they measure by the difference of intensities every few days. Also, they use UV light because they want to produce a mutagenesis, and they will measure the growth, the cell growth with light and without light. In this case, we can watch the distribution of the wheels with 3 different sizes. In this case, the intensity of the chlorophyll was higher in the smaller well. They determined the higher growth of microalgae in the biggest well. But according to the graphic, the growth rate was higher in the smaller wells, the growth rate. And they explain that in this case the, the shape of the well, because of this shape, the microalgae starts to grow more in the smaller size of well. According to related to the lipid content, the intensity of the or the highest content of the well of the lipid was you can see it in the shortest well. With UV light that they use, determine the most efficient exposure time of the microalgae to UV rays. So in this case, we can pass this part just to explain. We can watch the exposure of the UV to microalgae, in different periods of time. In this case, the most efficient exposure time was 5 minutes, as we can watch in this graphic. And it means that the microalgal growth increasing with this 5 minutes of exposure, and also the lipid content was higher. Also, they determine the influence of the light in two different areas: the region with light and the region without light. The algae in the region exposed to light show a cell grow higher than the algae in the unexposed light, as we can see in this graphic. According to the results that we already watched, in conclusion, the hanging drop microfluidic device developed in this study is optimal for measuring some important parameters, as the yield, the growth rate, and the lipid content in this case. Also, they could simulate the functions of a bioreactor with of course the necessary conditions for growth. And they determine that it's easily accessible device. So it allows the selection of microalgae for the production of biofuel, and it's able to be replicated in other studies. And they recommend that for future studies, use another varieties of microalgae to determine the most important parameter in the path.